Right now, your body is bombarded by massive amounts of physical data. Millions of photons bounce off urban landscapes and crowds, streaming directly at you. Simultaneously, invisible mechanical waves of varying pressure rip through the air, generated by physical impacts, like hands striking drums in a local performance. Add to that the airborne chemical data floating into your airway, molecules carried in the smoke lifting off roasting maize. Yet here is the biological paradox. The organ that actually perceives and understands all this information, your brain, never directly interacts with a single photon, sound wave, or molecule. In reality, the brain is locked entirely inside a dense cranial vault. It sits in absolute darkness and silence, physically isolated from the external environment. To bridge this gap, the body deploys highly specialized hardware known as receptor organs. The most complex of these are the eye and the ear. The primary function of these organs is surprisingly simple. They act as physical funnels, designed to catch and gather raw environmental energy from your surroundings. Once gathered, they perform a critical process called transduction. They convert that physical energy, light and mechanical force, into electrochemical nerve impulses. This is the only language the brain can read. Without these biological transducers successfully making that conversion, the outside world would remain completely invisible and silent to your nervous system. To understand how this conversion works, we start with human vision. It relies entirely on capturing electromagnetic light waves radiating from the sun or artificial sources. This diagram shows the horizontal cross-section of the human eyeball, illustrating the anatomical architecture required to capture light waves. Light first strikes the cornea, the transparent curved layer at the very front. Its shape acts as an initial lens, refracting incoming light beams. Immediately behind the cornea sits the pupil, a dynamic mechanical aperture dictating exactly how much refracted light proceeds deeper into the eye. In intense, bright conditions, the iris muscle quickly constricts to a pinprick, limiting incoming energy to prevent interior cellular damage. Conversely, in low-light environments, the pupil smoothly dilates to its maximum diameter, maximizing the number of photons it can capture from the dark surroundings. Once regulated, that stream of light passes through the pupil and strikes the internal lens. The sole physical function of this internal lens is to fine-tune the focus of the light beam, adjusting its trajectory so it lands perfectly on the rear wall. The entire front half of the eye is purely a mechanical targeting system, designed to deliver a clean, sharp optical signal to the biological transducer. Looking at the back of the eyeball section, we find the retina. This light-sensitive inner lining is where the actual biological transduction takes place. The retina contains specific photoreceptor cells. First, there are rods, which are highly sensitive and function primarily to detect shapes and movement in dim light. Second, there are cones, requiring much more light to activate, and specialized to detect distinct color frequencies. Once these photoreceptors fire, those signals pool together and travel directly to the brain via the optic nerve, shown exiting at the lower rear of the sphere. But this physical exit creates a structural compromise. Where the optic nerve attaches to the retina, there is no physical room left for photoreceptor cells. We can map this missing data with a simple test. Draw a cross and a circle on paper, spaced 10 centimeters apart. Close your left eye, stare only at the cross with your right eye, and slowly bring the paper closer to your face. You will hit a specific distance where the circle completely vanishes. The optical image of the circle is still entering your eye, but it has landed squarely on the optic nerve disc, where no receptors exist. Human perception is a constructed image. You do not usually notice this blind spot because your brain constantly works in the background, using contextual clues to paint over the gaps. Shifting to the auditory system, the stimulus changes completely. Sound waves are not electromagnetic radiation. They are literal mechanical variations in air pressure. This cross-section details the ear's internal architecture. The pinna, labeled A, acts as an acoustic funnel to gather dispersed sound waves. These waves are directed down the ear canal, marked B, colliding directly with the eardrum at C, causing it to vibrate. This vibration enters the middle ear, an area entirely dedicated to mechanical amplification. Resting against the eardrum is a chain of 3 tiny bones called the ossicles. As the eardrum moves, it sets these bones vibrating in sequence. This is a critical physical transition. Acoustic energy transfers from a large area of moving air into a microscopic area of solid bone, concentrating the kinetic force. If ambient sound waves hit the inner ear directly, they would simply bounce off. It requires this mechanical bone amplification to generate the brute physical force needed to push energy into the dense liquids of the inner ear. With mechanical force amplified, vibrations breach the internal barrier and enter the inner ear system. Auditory transduction occurs inside the cochlea, the fluid-filled structure labeled H. Fluid ripples inside the coiled tube bend specialized hair cells, firing off nerve impulses. These new impulses exit the cochlea and travel straight up the auditory nerve to the brain. A secondary mechanism is at work. The inner ear houses an entirely separate sensory system. Directly above the cochlea sit the semicircular canals, labeled G. These interlocking loops map spatial orientation by tracking fluid movement. This explains why an inner ear viral infection often causes a sudden inability to walk straight. If fluid in these canals becomes inflamed, your brain receives corrupted spatial data, destroying your balance. The structure we call the ear is actually two distinct biological instruments sharing a single entrance. They work simultaneously, constantly supplying the brain with the data required to hear a voice and the physical coordinates required to stand up. Despite detecting entirely different forms of energy, both the eye and the ear utilize the exact same biological strategy: gather, focus, transduce, and transmit. The architectural parallels are direct. The eye receives light, the ear receives sound. The retina is the receptor site for vision, and the cochlea for hearing. Lastly, the optic nerve carries visual data, precisely like the auditory nerve carries acoustic data. Tracking the linear progression of sound proves how dependent the entire sequence is on physical mechanics. The mechanical phase starts when the pinna funnels vibrations to the eardrum, transferring kinetic energy to the ossicles for amplification. The transduction phase begins when that force enters the cochlea, converting into electrical impulses that travel up the auditory nerve to the brain. Because these systems rely on microscopic symphinous cells, they are highly vulnerable. Extreme acoustic trauma can physically shear the hair cells inside the cochlea, causing permanent damage to the transduction sites that the body cannot repair. These delicate, irreplaceable structures represent a massive evolutionary achievement. They are the only biological windows through which your completely isolated brain will ever get to experience reality.